How did this genomics, the avian genomics project get started?
Yeah, kind of like the avian nomenclature project. My motivation for this and leadership of it began in a similar manner. That is, I am interested in understanding evolution of vocal learning and mechanisms of vocal learning. And I do a comparative approach, looking at the species who have this ability and those that don't. And there are 3 bird groups and 5 mammal groups that we know of so far, and or at least consensus. And so basically, in order to search for the genetic underpinnings of this trait in multiple species, you need to have genomes. And so, but I didn't have the money to do it. And so what I discovered is that lots of people want genomes, and there are lots of bird fanaticists out there, including in the sciences, that want the genomes of many species, like to solve the family tree of birds. And I was also interested in knowing if the vocal learning bird groups, hummingbirds, songbirds, and parrots, had a common origin or a independent origin. We always thought independent, but the brain pathways for vocal learning were so remarkable that a lot of people were saying this has got to be common origin. And so I got involved in the project to sequence the genomes of 48 bird species that became the Avian Phylogenomics Project, representing all bird orders, including vocal learners. The folks who were supposed to lead the phylogeny effort were moving a little too slow for me. So I kept pushing things along. And then I ended up becoming the leader of this phylogenetic effort, even though I wasn't the phylogeneticist. It turns out vocal learners, we generated a genome scale tree of all these birds, and they are independently evolved. Then once we had those 48 species, I thought I can stop there. I had close relatives of vocal learners, but it turns out a lot of these genome assemblies were fragmented. They were generated with these short reads that don't allow you to go through long stretches of duplicated sequences, and they don't also read through GC-rich regions as well. All right, and so we were having difficulty doing functional studies in birds because we had bad genome assemblies. At the same time, leadership of an organization called the Genome10K organization approached me and said, Eric, can you do with our vertebrate group to sequence 10,000 vertebrate species what you did with the birds? And they basically threw the leadership in my lap, and I became chair of the group. There were 3 leaders before, and now it's down to 1, this new person, myself. And they were a generation older than us. I think they were trying to pass on the ball to the next generation. And so with that, I decided to do 2 things. The first was say, why stop at 10,000 species? Let's make our mission all 70,000 vertebrate species on the planet. And because it's a lot of effort to do 10,000, I don't think it's as much more effort to do 70,000. That's what I was thinking. Then the second is, I can't deal with these low quality genome assemblies anymore. So we worked with the sequencing companies, and I brought together an assembly team of experts, from various places around the world, to work on sequencing technology and genome assembly technology for the last 5 years to get the best genome assemblies possible. And we just published a special issue in Nature a few months ago on the first set of genomes, including starting out with the hummingbird. It's on the cover of Nature, because that's the first one we started with, one of our vocal learners. And now we have the best genome assemblies out there. And we're getting the regulatory regions of genes that are specialized in song and speech circuits we did not see in the old assemblies. That's causing genes to come up or down in the speech areas. And the model that we put in place for these high quality assemblies and a phylogenetic scale of doing genomes is now being adopted by major genome consortiums that are starting to sequence all eukaryotic species on the planet, like all 2 million, called the Earth Biogenomes Project. And to redo the human genome over again, a pan-human genome reference, 350 people from around the world, representing 95% of human diversity, with very high quality complete genomes.